Kia ora koutou katoa and welcome. Thank you for joining us in this session of the Thriving Cities webinar series. My name is Rachel Nepia and I'm the Program Manager for People, Cities and Nature, and I'll be your host for today. This webinar series explores what it means for cities in Aotearoa, New Zealand to create thriving biodiversity and healthier urban environments for people and for nature. So today's session is going to explore emerging research on airborne and soil microbial communities as mediators of urban restoration and public health outcomes. To help us through that research, we are delighted to welcome Professor Martin Breed, who is from Flinders University in Australia. Martin examines the role of microbial and genetic diversity in ecosystem function and restoration. His research uncovers the often overlooked microbial underpinnings of healthy urban environments. With him today, we have Dr. Greg Jacobson from the University of Waikato. Greg's research focuses on soil microbial ecology in restored and degraded landscapes, investigating how microbial communities contribute to ecosystem resilience in urban settings. Together, Martin and Greg will share some research on aerobiomes and soil microbiomes across restoration gradients in New Zealand cities and correlations involving the microbiomes of New Zealand's urban lizards. After their presentations, we'll have time for questions. So please use the Q&A function anytime during the presentations, and we'll get to your questions at the end. So first up, welcome to Professor Martin Breed.
Wonderful. Thank you so much, Rachel. And hello, everybody. I'll just share my screen and get started. Okay. Can you see my screen? Very good. So thanks very much for the invitation, Rachel. Delighted to talk to us all today. And I'm going to be covering a bit of a different topic than perhaps what I was originally asked, but I think we'll get there in the end. So it's all about urban green spaces and these two things, bioaerosols and the aerobiome. And just so we're all on the same page, bioaerosols, these are the particles originating from living organisms in a given airspace, and aerobiomes are the communities of microbes in a given airspace. And so as Rachel described, I come to you from Australia, from Ghana land in Australia, and pay my respects to elders past, present and emerging. And just very briefly, a little bit about me. So I'm a professor here at Flinders University, run a group called the Frontiers of Restoration Ecology Group, and we work on a lot of areas that relate to restoration. And so restoring climate resilient ecosystems, including provenancing or eco-sourcing, a lot of soil microbiome work, a lot of genomics, and soil ecoacoustics is a new toolkit we're getting access to, as well as restoring urban green spaces for human health outcomes and different ways of monitoring that. And also I do a lot of teaching and teaching coordination at Flinders University. So I really enjoy that teaching research nexus, and there's some components here that I think could be integrated well into teaching programs. And of course, this isn't just my work, this is a work of many, and I just want to acknowledge these people straight away. So the first group is the research aim that I help coordinate through the People, City and Nature program, and the people are described there. But a lot of the aerobiome and bioaerosol work that I'm going to present is work jointly and oftentimes led by Dr. Jake Robinson, who's at Flinders University as well, but is a prolific writer. You can check out his books and YouTube channel here. So thanks very much for all of my co-authors and co-contributors. So firstly, about bioaerosols. There's some really interesting and important fun facts. So did you know that we inhale thousands of pollen grains each day, but we also emit millions of biological particles every hour? In a cubic meter of outdoor air, this typically contains 10,000 to 1 million bacterial cells. Urban dwellers like me typically inhale millions and millions of bacteria each day, which means we are in this very biologically rich air environment when we walk around. And why is that important? Well, historically, bioaerosol research was focused on the unhealthy things. So we're talking about pathogens and allergens. But together with Jake, we put forward probably a provocative idea, but we wanted to push the boundaries to make sure that people who are interested in bioaerosols thought also about the things that were good for us in the air. So good air quality isn't just the absence or minimization of things that are bad for us, but also a safe level of exposure to things that are good for us. And I'm going to unpack some of those in a tick. But these includes the microbes in the air and things like phytoncides, these volatile organic compounds that are released by many different plant species. And don't have a lot of time to get into the details, but the principal understanding of the role of microbial biodiversity and the exposure of us to it is fundamentally important. And this is acknowledged through this image here on the left-hand side, through the World Health Organization and the Convention on Biological Diversity. So they see biodiversity as the root of health outcomes, and one of the principal avenues is through microbial biodiversity. And one of the clear pathways of that health effect is through training our immune system. So our immune system is related to our exposure to microbial biodiversity. And I'm here going to present why the aerobiome then matters. And so this is work done by Jake Robinson, myself, and this is my lab tech Christian out in the field doing some really high-tech research with tomato steaks and Petri dishes, measuring the aerobiome. And I'm going to skip quite quickly through to some results. So soil microbiomes supply the aerobiome, and you can see it here on this plot on the left. The diversity of different bacteria in soil was significantly higher than that up in the air, but if you're a small child, you get exposed to more rich aerobiome than a taller adult, let's say. And this has really important implications for how we as people interact with our environment and get exposed to this microbial biodiversity in the air. And let's go on. So we get colonized by this aerobiome diversity. And so here's just one study where there's a lot of complex things happening on these plots, but basically I've circled the pre and post exposure. So this was only after half an hour to 45 minutes of exposure to urban green spaces. And these plots show the change in transitions of the community of nose and skin microbiomes on people in these environments. But this here shows an elevation of diversity after exposure to the urban green spaces for, as I said, only 30 to 45 minutes. But what's really important is that we can actually affect what community of microbes are in the aerobiome, and we can do that through restoration interventions by affecting the vegetation community. So again, this is a plot that shows from low to high vegetation complexity having an effect on at least the alpha diversity and also the composition. I haven't got it here, but also the composition of the aerobiome in urban green spaces. So you can see in the plot here, this is a map of the city of Adelaide, well, a very small part of it. These are the parklands in the southern part of the CBD, and this is where we were doing our sampling, showing, yes, vegetation complexity affects the diversity of microbes we're exposed to, but also the proximity to things like trees. So the closer the aerobiome sample was to trees, the richer the diversity of microbes in the air. And without naming individuals, we'd love to have more work to present from the New Zealand cities, and I tried really hard to get the results for you today, but I don't. So really what we're doing in the PCAM project that relates to what I've just described there is really trying to drill down to some of the mechanisms at play. So we've got a lot of interest in the leaf litter. That's a very important, rich, biodiverse layer of forest ecosystems. And so we're looking at the vertical stratification of the aerobiome. So how does the soil seed the leaf litter that seeds the air? And not just bacterial diversity, but fungal diversity. And we've asked this question across 3 New Zealand cities. And so unfortunately, you'll need to wait a little while until we have the results to come back. They're just not quite ready to share, but please keep note and track our research as it comes out. We will release preprints as soon as they're available, and there's some exciting results coming. and some other future research. So how would one set up these monitoring stations in urban green spaces? Well, we can't have petri dishes and tomato stakes everywhere, but we do have a very large number of spider webs. And it might sound like a bit of a wacky left field idea, but spider webs have been shown to be really good proxies of the mammal community in certain environments. And we've got a study that's been undertaken right now to understand whether spider webs can be these natural aerobiome samplers that we could be sampling in cities to get an understanding of what microbial diversity is in the air. And some of the other research that Jake and I have sought funding for is to utilise some of this research and bring it into childcare settings. So there's a lot of air conditioning. It's a very hot and dry environment in Adelaide, and we're looking to build in some of this biodiverse exposure into childcare settings, utilising planting arrangements around air conditioner units in how that can be utilised to manage and improve the air quality in childcare settings. So I've just skimmed the surface. I need to give a lot of acknowledgement. So thanks very much for the time. I've just gone over my 8 minutes. So very much welcome any questions you may have. Chuck it in the Q&A or send me an email and I'll get back to you in good time. So that's the end of my presentation. Thanks very much.
Thanks, Greg. That was so fascinating. We'll turn the time now to Greg.
So I'm Greg Jacobson, and my role in this project, working on the pecan work, was to study the a population of New Zealand lizards and 2 different species of lizards to try to ascertain the amount of connectivity between the soil microbiome and the animal microbiomes. And we work closely with Nikki and Kat in Wellington and Chris to carry out this study, and I'll go into some details on what we were interested in looking at and the species we focused on. So our main study questions were what bacterial communities were found in lizards and soils that these lizards lived on, and with the similarities and differences. And then we also moved on to look at what lizard-associated communities were linked to the local soil communities. There's some crossover between parts 1 and 2 there. And then trying to correlate the bacterial or microbial communities within the lizard and on the lizard with the lizard's body condition, which is a measure of health. We picked 2 species. So these are species which are found around Wellington in New Zealand. And so we've got the northern grass skink and the Rakawa gecko. One's more nocturnal and one's diurnal. They have a diet of insects, and they're a bit omnivorous. They eat sometimes nectars and small fruit as well, but primarily they survive on eating insects. And they live in coastal habitats around Wellington. In this study, we trapped and collected 17 of the skinks, so the northern grass skink, and 7 of the geckos. So they're both lizards, but skinks and geckos are under the classification of lizards. So collection and trapping was via these pitfall traps. Animals were sort of humanely captured and alive captured into pitfall traps, which does them no harm, and they just detain for some time while we sample their microbiome via little microtip, like cotton buds on the end of a stick, basically. And then we also collected stool samples or poo samples from the animals, which were housed overnight in the lab in sterile housing. So we were able to collect microbial communities or assess the microbial communities within the stool sample and on the skin swab or cloacal swab from the animal. We measured the length, we weighed them, and we were able to determine sort of a BMI equivalent for animals, if you like, the body condition index, which is a function of their length and their weight. And it's very analogous to the BMI in humans. We collected DNA from those samples, and from that DNA we were able to determine what bacteria are present, and to go into some studies on the composition of the bacterial communities and so forth, which I'll go into in the next few slides. This is a plot of the types of bacteria, if you like, at the sort of a higher family level, not down to species level, but it shows that soil is a very sort of a richer and more diverse sample or substrate than the animal samples, which were cloaca and stool. Cloaca and stool were far more similar. These are the color here in this, what we call a heat map, indicates the relative abundance of each of these phyla or families of bacteria. So our finding there was that the soil microbiome is very distinct from the other two. So it's sort of maybe common sense to think that maybe the soil is going to be far more diverse, meaning having more species and a more even distribution of those species than there's animal samples, because the animals will have inbuilt ways of filtering for useful bacteria, maybe physiological filters that just select those bacteria that confer some health advantage to the animal, but it's not something we delve deep into in this study. Next, we're interested in seeing whether the animals had some of the organisms, or there was sharing of the organisms between the soil and the animals themselves. And to do this, we did a computational study using indicator species analysis, where we look at soil and get a fingerprint of the different species which are characteristic of that soil. And it worked. We had a significant sort of population of bacteria or groups of bacteria, clustered groups of bacteria that were distinctive for site 1 but not site 2, which just because we collected a lot less samples at site 2, so this worked well, but we're focusing mainly on the site 1 data here. So the fingerprint is like the control, and from the fingerprint we can then look at animal samples and see whether they were enriched for the fingerprint that was associated with the site they were collected from, which would indicate some transfer from the site soils to the animals themselves, as evidenced in the stool and cloacal samples. And we had definitely an association where animal samples, both the stool and the cloacal samples, were enriched for the fingerprint of the site they were at, which is a good indication of some transfer or acquisition of those bacterial species from the site. And it's nice to see some actual validation of that effect using that fingerprint type analysis. So the site 1 lizards were confirmed to carry that site fingerprint. We're also interested in just doing an analysis of whether bacterial diversity, that's number of species and how evenly they're spread in the species are spread across the sample, with a health metric of our measured body condition index in the lizards. For a subset of the lizards, and we've split by species type. Remember, we had 2 different species, the gecko and the skink, and by gender as well. So when we split it into subsets, we saw a sort of emerging pattern of an association between increased diversity and better body condition, or greater body condition by this index here. So that indicates more diversity in the bacterial population, in this case in the cloaca. We didn't see it in stool, interestingly. It does associate with a better, higher body condition index, which is sort of a proxy for health. It's not perfect for it, but it's so this is a very small data set of only 6 animals in this case. So this is more exploratory pilot data, but it's an interesting effect which we'll hopefully explore later and in bigger data sets, so more animals. Oops. Okay, so in conclusion, so we know that the lizard microbiomes are very different from those soil microbiomes, which you saw in that heat map. and the other experiments showed there was, or the other analyses showed there was traces of the soil bacteria in the lizard samples. We saw that with some sharing of organisms across that heat map as well. And then we also showed evidence that high microbial diversity in the cloacal samples was associated with better health. So that's, and also that animals did retain or acquire and retain some of the organisms present within their local soils that they have this intimate association with as they live directly on them. So those are the main points for me. And so just to emphasize, we need to consider the microbiome and within these habitats as an important contributor to animal health and potentially the health of any animal, including humans, interacting with those environments. And that's all for me. Thank you.
Thank you, Greg. And thank you, Martin, also for your comments. It was incredibly interesting. The time really flew while you guys were speaking. It's time for questions now. And we've had a few in the chat that mostly correlate with restoration of urban environments and applications for your research. What can we do to improve microbiomes and aerobiomes in those places? So if you have any insights that you'd like to share on that question, would be appreciated.
Yeah, so as per the chat, it's been some really good questions going down there. I guess my general sense is if you're after local knowledge and local fungal species and strains, particularly in natural ecosystems, find someone local in New Zealand. I'm not based there. I will hesitate to give direct information there, but I'd certainly say there's very strong evidence that the soil microbiome responds quite slowly to restoration plantings, and a lot of that is driven by the fact that the soil remembers. And so I put that in the chat. But what that generally means is if there's been agricultural inputs for, 100 years, you can't just plant some native plants and expect the soil health to recover naturally. There probably needs to be either another 100 years to wait or some quite strong interventions would be my suggestions. And I've added a couple of links to some papers that might get to some of that in the chat. And yeah, happy to respond to more specific queries if they come up, and I can't quite talk and read at the same time, so I'm struggling to find the next question, but maybe I'll pause from speaking and wait for additional questions to come through. So, or Rachel, if you could field more questions, feel free. Thank you.
Okay. One thing that I thought was really interesting in your talk, Martin, was about that connection between the air quality and the land quality, and I just wondered if you have any insight as to whether people who are looking at those different environments are talking, and how we can maybe foster a bit more of that so that they can work together.
Look, I think it's a conundrum. So people who think about air quality often sit in EPAs or health departments, and the people that are concerned with soils and native biodiversity tend to be in environment departments. So how does one build that conversation? Well, probably requires us to do some research, which is great to demonstrate use cases, but creating forums, I guess, to some degree, exactly like this, to have those conversations, because oftentimes those are siloed environments where health and environment don't work together, and that's a real problem. So increasingly science is certainly thinking about trying to be more interdisciplinary, but I get the sense that the community, as well as government departments, government agencies, are increasingly aware of the complex and connected nature of things out there. So I'd say it's up to us to be good communicators and not be afraid to not know about things when we're in the room with a diversity of audiences and make some use cases. So I'd say keep going with conversations like People, City and Nature created, where we do have that diversity of stakeholders and perspectives working together. But yeah, it is by no means easy, and I think we need some really good demonstrated use cases to show or demonstrate the why. Why would we want those two different sectors to work together? And I think we've only scratched the surface here. Yeah. Robin's got a question about inoculations. Can I yes. Go with that one? Cool.
I would love you to do that. Thank you, Martin.
So yeah, Rob, like a really good point. And I'd say if we talk about inoculations traditionally, they probably come from the agricultural sector, where single strain, single species type of work is the dominant force. But as an ecologist, I certainly would argue that the other way of going, thinking of whole communities, in fact, working with the communities of the microbes or the other things in the soil together with their habitat, is probably the most effective way to do inoculations in an ecological setting. Because if you throw microbes into a soil like we might do in our backyards, there's all sorts of things waiting to gobble them up or push them out or not let them move into that habitat. So actually working with the habitat, as in the soil, the leaf litter, the carbon sources, the other nutrients available, if those things aren't right, why would the microbes want to establish? And so I very much agree with your sentiment of thinking about it as an ecosystem rather than a single strain or a single species based inoculation. That's agricultural. There's a purpose for that, obviously, but ecologists need to think differently, which we are doing.
Do you have any comments on that, Greg?
Yeah, I was buried in the questions as well. Inoculations, I agree that I agree it's important to try. It's not something I've had a lot of experience at. I'd rather not comment in too much detail there, but I think certainly if it's true of the human gut, it's always the case where higher diversity is always associated with better health, and those bad health things in the human gut, for example, which is one of these microbial environments, is always bad health outcome situations like metabolic syndrome and diabetes even and so forth is usually associated with lower diversity. So in the very simplest terms, increasing diversity at any of these sites must be good for inhabitant species, I guess.
Yeah. And we could extend that a little bit with some of the methodologies. So Maria picks up on the point that a lot of the compositional or the species-based microbiome work is limited and has emphasized the importance of function. That's a hard agree, absolutely. And so richness in different species of microbes certainly doesn't correlate with richness in numbers of microbial functions, for example. And I want to add a link to, so Craig Liddicote's part of People, City and Nature, and he's given a talk, I think, in one of the earlier seminar series. Is that right, Rachel? So maybe linking to his talk would be useful, but I'll add a link to his preprint where he's pushing the boundary beyond just function, actually asking the question of what can the microbes produce from a compound perspective, and understanding functions. Just because there's a gene for a function doesn't mean that that gene is actually activated, and so we've got to think beyond function as well. And so that's a very fruitful area of research that we're tackling, but many other groups are doing as well. And there's no set response in how to appropriately measure a microbiome, I think. So I very much agree with your point, but that's an area of development in the discipline. So keep going. Yeah, exactly. So transcriptomic work in soils. We had a PhD student, Stella Bruckman, who's got a paper, I think, under review, looking at the correlations between the metagenomics and the metatranscriptomics data from soil environments. And there was some correlation, but quite a bit of heterogeneity was captured by the metatranscriptomes that was not present in the metagenomes, based exactly, I would imagine, what you're thinking about. So the expression profiles being very different from the gene, functional gene profiles. If only her work was published. It will be seen.
Awesome. Thank you, Martin. And I've also just shared the link to that webinar that you mentioned, which addresses Maria's question about functions of those microbial communities with a specific reference to urban restoration. So that one's really interesting if you've got time to have a look at that. And that is about all the time that we have for questions for today. It went so quick. That was fascinating. So thank you again to Martin and Greg for sharing your research and insights with us today. It has really got me thinking. I hadn't even considered air conditioning and indoor microbiomes, and yeah, this just really opened my eyes on that topic. So thank you also to everyone that joined the session today. The recording will be up on YouTube in the next week. So if you want to share it with anybody who you think should have been here today, you can do that. We hope that you've learned something and got a new perspective here also. Our next webinar is in July, and it will introduce a kaupapa Māori tool for assessing restoration projects, and that will be with Dr. Sean Awatiri and Dr. Irina Walker. So keep an eye out on our website and socials for the registration link. And until then, kia pai tora. Have a great weekend.
Thanks, Rachel and everyone.